We're in part 2 now of the neural tissues, and we're looking at page 391. And as you can see here, we're in chapter 11. Here's an electron micrograph of a multipolar neuron, and here is the classic multipolar neuron. It turns out about 99% of all neurons have this configuration, which is lots of options for incoming information here. So it's possible for us to have a lot of associations with other neurons. And in fact, that's what maturation is the development of many associations with neurons of other provinces of the central nervous system. Okay, so, and then of course we have the inflow, which is sensory, and the outflow, which is motor. Okay, so this is our previous episode here. We took a look at this, and I noted to you that there are movements down the axon. And in our next page over here, we take a look at the developing Schwann cell. Do you see it? It's beginning to wrap itself around an axon or any other what we would refer to as the white fibers. So let's just review that, gray and white. Basically, the gray matter is wherever we find the bodies, okay, the cell bodies, the major cytoplasmic portion of the neuron. And then wherever we have dendrites and axons, they're usually collected in areas that are referred to as white matter, gray matter, etc. Okay, and so we're going to wrap up the fibers because they can travel a long ways, as indicated before. They travel gross anatomy distances as microscopic entities, right? And so in order to make that distance over the microscopic distance, microscopic size of the structure over that great distance, we need sometimes to create this sort of little myelin sheath here. So here you see the Schwann cell wrapping. It's wrapping a little further around. Here it's wrapping quite a few times around. And then here we have an electron micrograph of an axon with lots of wraparounds. As you can see there, it's quite well protected inside of its sheath. And of course, there's going to be some connective tissue around the individual axon, referred to as neurium. So we'll talk more about that as time goes on. Here they're talking about transport along the axon. There's a great deal of movement away from the cell body, it says here, antegrade movement. And then the opposite also occurs, which is retrograde. Not quite as much of the retrograde persuasion, but nevertheless it happens. The transport in the antegrade position has to do with basically moving things to the axon terminal that you need there. So there's going to be various plasma membrane outer portions as well as mitochondria that you need to get down in there. And then, of course, there's neurotransmitters that are produced, synthesized in the cell body, and then transported to the axon terminals. And then there are substances that are transported in the other direction, in the retrograde. So if you need to pull back an organelle and repair it or degrade it in some way, metabolize it, you do that. That's probably the main thing that's done right there. Okay, there's also information flowing back and forth so that you can grow. You can send growth factors, it says here. And so there is a relationship, actually, and how shall I say, a interactive relationship that, in other words, you can touch something and develop a relationship. You can become more sensitive to touch. You can become, etc. You can read more about that sometime in the consciousness of the cell. I have a friend who did a master's degree in that before he went to medical school. Very interesting topic. At any rate, so there's a one basic bilateral transport mechanism. Appears to be responsible for the axon transport, uses ATP-dependent motor proteins. In other words, we're going to be using myosin and acting like molecules, only a slightly different variation. And we're actually going to propel things down through the microtubules, or up through the microtubules, as the case may be. And then, of course, there's diseases which utilize those mechanisms, such as polio and tetanus and herpes simplex, rabies, that kind of thing. So there are actually, these are viruses. Okay, so viruses can move around. There's even some suggestion that we might use viruses as a therapeutic method of cleaning things up. At any rate, the myelin sheath, very important. I was trying to get a place here where they mention the speed at which things happen. If it's an unmyelinated axon, the speed of the action potential down that, or the signal, we'll learn what that is, ultimately is only about 1 meter per second. If it's myelinated, it can get some really high speeds going, like 100 meters per second. So myelination really matters. And let's take a look at the different types of cells. We have a table over here on page 394. We have different types of cells. 99% are these, which are the multipolar type cell here. It's listed as multipolar. It's the most common class of cells. A lot of the cells throughout the brain are like this throughout all of the motor activities of the anterior and lateral horn, engaged in muscular activity, or smooth muscle activity, all that kind of stuff. Then we have a significant number of these cell bipolars, and what they do is they take information and then transfer it usually to a cell like this. Okay, so these transfer little bits of information over short distances. We use them in the retina, we use them in smell, olfaction, in a number of different places where we need to pick up the information, usually generated by generator potentials in receptors for various types of reception. Okay, so there can be visual receptors, there can be auditory and particularly sensory reception for smell and olfaction, that kind of thing. Then we have this one, which is pretty common also. And in this one, we have a central process and a peripheral process. And out here, we're touching things. Often is the receptive type of thing. It could be temperature, it could be touch or proprioception. Proprioception has to do with the position of a joint. I know if my hand is, or fingers are bent, or if they're straight, or if my head is tipped to one side. I can tell these things. That's proprioception. I can also touch things. I can understand the difference between velvet and the smooth surface of polished steel, right? And so that's information that comes in. We collect the information. We have the cell body that is not often in the central nervous system in this case. It's actually just outside the spinal cord, and we have a reservation for it there in a ganglion, we call it. Oftentimes when we have ganglions, that's a reference to where cell bodies live outside the central nervous system, and we use the term nucleus or cortex, gray matter, for where we have cell bodies inside the central nervous system. And then we have a central process which allows the information to continue on to the next part of the circuit, the next neuron, and so on. Okay, so they're referring to all of this as an axon. I tend to refer to just right here as an axon, or right here as a dendrite, and then right here as an axon. But at any rate, you can consider that for whatever you want. When I test you, I'm going to be saying that things leading up to the cell body is called a dendrite, and that things proceeding out beyond a cell body is considered an axon. So I'm going to be using that definition, which I think is a common one. Okay, then we have trigger zones. So it's possible to get an action potential, we get the signal, if we trigger this part of the cell. This part, not so much. There's a kind of there's a kind of thing that we do out here which prevents this from going forward, actually. So, but the actual trigger zone for what this neuron is supposed to do, it happens only in certain areas, the dendrite and the cell body usually, and not so much the axon. The axon is unidirectional away. Okay, and let's move on then to all of these other types of things. Basic principles of electricity we need to know next, and so that's going to be the next part of our discussion right here, learning about voltage.